So hi, hello and welcome again. Microbehunter here and welcome to another Microscopy podcast. Yes, that's a little bit of a new thing that I've started a few weeks ago. Besides YouTube videos and tutorials, YouTube tutorials, I decided why not uh, give it a try also with a podcast. So something that you can also listen to and not only watch. And for this reason, I of course have to choose topics that are suitable for audio only. And I've therefore decided to simply answer a couple of questions that you might have. Maybe I'm going to pick up on some different topics in the future as well. But for right now, we're going to stay with a couple of the questions that you posted either beneath the YouTube videos or that you sent me over email. The questions are on a variety of different microscopy related topics. Some are a little bit more practical, some are somewhat a little bit maybe more biological in nature. Yeah, but I hope at least that there is something interesting in it for everyone. I'm going to start off with the first question here, and goes like that. Have you ever mixed a bacterial sample with blood to see white blood cells destroy them? Is this even feasible? That's an interesting one. Okay, I need to give you a little bit of background information here. Is one of the videos that I've made, and which is actually quite successful, is a video which shows white blood cells, so-called phagocytes, moving around on a microscope slide and searching for bacteria. And it would be now kind of interesting that maybe it's possible if you are to add some bacteria, then maybe it's possible to actually see them eat up those bacteria. And indeed, this is something that is possible and is feasible. I have not yet tried doing that, but I will take this question here as a good, I would say, incentive or opportunity to actually try it out myself. And maybe this is also going to be a video in the future. Now, I think there are things that you have to take care are the following. First of all, of course, you need a source of bacteria. I do not recommend that you grow your own bacteria because for safety reasons, but also because the concentration would be too high. So what you need is you need only a couple of individual cells that you then can focus on under the microscope, and then you can observe whether white blood cells will actually approach it and engulf it to eat it up. Of course, there are also safe sources of bacteria, for example, yogurt bacteria and so on are of course available, and I would probably try it with those first. Now, I have some time ago seen in YouTube somewhere a video, or was it a YouTube short, where someone put a nematode worm, together with some blood, and it was then possible to actually see that the white blood cells attacking the nematode worm. So this therefore should be possible. And the reason why this actually works is because the bacteria, they're releasing certain chemicals, and the white blood cells, the phagocytes, are following the gradient of these chemicals and are therefore able to detect where they can be found. Now, if you actually were to grow your own bacteria, even if they're safe ones, and if you take a small sample of of a bacterial colony, for example, and mix it with blood, I do not think that this is going to work because there are going to be millions and millions of bacteria in a extremely small sample, and for this reason, you have to dilute it down, of course. And then the concentration of the bacteria goes down. But at the same time, you have to be careful that you do not add too much water to your blood sample on the microscope slide, because otherwise there is the danger again that the blood cells, the red blood cells, and maybe also the white blood cells, that they will die because of an osmotic imbalance. The water will go into the red blood cells and they will pop. And in order to prevent that from happening, you have to, of course, also add a little bit of salt to the water. It's usually a physiological saline, as it's called. It's a 0.7% salt water solution to make sure that the bacteria are diluted in an environment which does not then harm the red blood cells and the white blood cells when you mix it with the blood. So you see, it is possible, but there is a little bit of preparation work necessary as well. But generally, it should be possible to see essentially the white blood cells engulf and eat up and remove the bacteria. So, but I think this was a very technical explanation right now. But I think I'm going to try it out, and then maybe I'm able to report back on this later on after I've tried it out myself. I would like to move on now to the second question. The question is a little bit longer here. Actually, there's two parts here. I have two questions for you. First, during your live streams, you sometimes mention that you avoid using oil immersion for fear of accidentally running other objectives into the oil. Why not simply remove the other objectives first to prevent this from happening? Removing them doesn't take much time and could save a lot of trouble. Yeah, and then there's also a second question which I'm going to read later. So a little bit of background information here. In the case of oil immersion microscopy, what you have to do is you have to place a drop of immersion oil on the microscope slide, and then the oil immersion objective, that's usually the 100 times objective, is directly rotated into the oil. This means that the objective becomes essentially contaminated with oil, and it's specifically manufactured like this so that the oil is not going to do any damage. You, of course, later on you want to use some tissue paper, some lens paper rather, not just regular tissue paper, but you should be using lens paper to remove the oil. However, there is, of course, always the danger that when you're changing the magnification around it, once there is already oil on the microscope slide, if you use now, let's say, a 40 times objective, which is a non-oil objective, that this 40 times objective also becomes contaminated with oil. And if this happens, then this is not good, and might even be able to damage the objective, because in many cases those objectives are not oil tight. So this means there is a possibility for immersion oil, to actually enter the objective itself. So basically it will find its way between the objective and the lens of the objective, the front lens of the objective, and kind of go into the objective. And that's something you want to avoid. So the question is why don't you just remove those? Well, this is not practical. And the reason is because I need the lower magnification objectives to find the focus. Because I need to start with a 4 times objective and then focus it, center the slide, work my way up, and only after I've used the 40 times objective and then I switch over to the 100 times oil immersion objective, only then will I add immersion oil. But then I know exactly at which place on the microscope slide I want to look at. And if I remove now the other objectives, then essentially how am I supposed to find the focus when using the immersion objective? Yeah, I need the other objectives to actually position the specimen and do all of the preparation work. So this is a little bit of an issue. Now, some microscopes, they have a so-called a focus lock lever. This basically means that once everything is in focus with the oil immersion, then you're able to flip over the lever, and then this kind of the microscope gearing system will remember the position, and then this means I can use the coarse focus knob to lower the stage, and then I can add the immersion oil, and then I can raise the stage again, and then it will move up to that point that basically it was remembered, right? So that would indeed be a possibility, but again, this is also not practical if I do not have any other objectives present. Yeah, so because I need those objectives again actually to center the next microscope slide. So this is a little bit unfortunate. One just has to be a little bit careful, but I'm just talking out of experience, especially when you're using the microscope for many hours like I'm doing, and sometimes you simply forget, or I forgot once that I was using the oil immersion objective, and I simply rotated the 40 times or 60 times non-oil objective directly into the immersion oil, right? I completely forgot about it. And this is because I'm not always looking on the side. I'm just looking through the microscope, and then I'm rotating I'm changing magnification, and then I simply forgot about it, right? So this can happen quite easily. But unfortunately it's not practical. So there's a second question here. My second question is about refurbishing an Olympus BH2 microscope using separately sold parts. I'm very mechanically inclined and was wondering how difficult it would be to salvage parts from other non-working Olympus BH2 microscopes. Well, this is definitely a possibility, and this is actually something that is maybe not commonly done, but it is done. right? Those brand microscopes from the 1970s, 1980s, like the Olympus BH2 microscope, were extremely popular microscopes, manufactured in quite large quantities, not only from Olympus, but also other brands like Zeiss, the Zeiss Standard, for example, had highly modular designs and with a lot of fairly large second-hand market, therefore, and it is indeed possible to put your own microscope together. However, and that's maybe also important, it does not mean that this is going to be cheaper, right? This way, because you might not find the parts that you need, so you have to buy yourself a second microscope, and then you basically disassemble it and you reuse the parts. So you see, it's not automatically cheaper this way. How can it be justified? Well, it can be justified because you're learning a lot and because it's interesting and it's fun to do, right? And indeed, there are lots of people, I wouldn't say lots maybe, but several people who like actually repairing old microscopes and refurbishing them and then reselling them again, for a higher for a higher price, because then it works. And the good thing about those microscopes, those brand microscopes, is that yes, they were actually designed to be serviceable. This means that there are service manuals that you can download and obtain, and every now and then, every couple of years rather, the microscope should be disassembled in any case to clean the gearing system and to remove the lubrication oil, and because over the years it becomes solid and it starts to increase in viscosity, and then you cannot turn the focus knobs anymore. But not a problem, because essentially there are service manuals available which allow you to disassemble a microscope and then to actually get it cleaned and fixed again. But you don't even have to go to that detail. If you're able to get eyepieces and objectives and whatnot, right? You should be able to put together your own microscope. Now, another thing that I want to mention is that the BH2 microscope, just like many other traditional systems, essentially had 160 millimeter optics. And yes, there are modern microscope optics also available, fairly low cost, that you can actually fit on the BH2 microscope. The BH2 from Olympus was not an infinity microscope. And for this reason, you can still obtain new objectives that would actually fit. So you do not have to rely in all cases on original parts, even though that is, of course, also a possibility that you can do, right? So I would say you can justify refurbishing old microscopes, specifically then if you have a specific interest in doing so. But you have to be aware that it might take some time for you to obtain all of the parts. It's really a question of where do you want to place the focus. Do you want to place the focus more on the hardware part, on microscope maintenance and refurbishing, which is certainly very interesting, or is your focus mostly on observing of nature, making videos and pictures and so on? It really depends. Different people interpret the hobby differently, or combination, of course, of both. And this is essentially something that I would say that it's possible. Now, I know someone who once emailed me several years ago who actually was able to convert a BH2 microscope to DIC, to differential interference contrast microscopy. This actually did cost several thousand euros, even though all of the parts were essentially second-hand, still cheaper than if you were to buy it maybe new. But you have to understand that those are highly specific parts, which might be a little bit difficult to get, and which were not manufactured in so large quantities. So this can actually mean that depending on what you want, it might not quite be as cheap. I remember a couple of years ago, I converted an Olympus CHA microscope, also a microscope from the 70s and 80s, for phase contrast, right? And essentially, simply the phase contrast condenser cost me around 300 euros, maybe it's somewhat over 300 US dollars, right? So you see, it's not that cheap in upgrading it, but it is certainly something that if you're interested in, you can do. So I would like to move on now to the next question here. So by the way, these podcasts are amazing. Thank you very much. Okay, I have a question for a future episode. Do microscopes exist that have a vacuum between the objective and the slide? Maybe it doesn't have to be a full vacuum, but some kind of a sealed space to prevent outside air and particles from interfering. Any insight would be appreciated. I'm sure something like this exists, but I'm not sure what it's called. Okay, I will assume you of course I assume that you're talking about light microscopes and not electron microscopes, because in electron microscopes you have to place the sample in a vacuum in any case. So it's not possible to use moist samples that contain water, because the water would immediately evaporate and fill the chamber, and it wouldn't be a vacuum anymore, but there would be water vapor in there. But for light microscopes, I'm not aware of anything like that, because essentially the objective itself would then have to be sealed airtight around the specimen, and this is actually not something that is normally done. And why would one do that? The air basically does interfere because of a refractive index, but it's basically the same as the refractive index of a vacuum. So there is actually not, concerning refractive index, there's not really a huge difference. And honestly, the microscope objectives are already designed like this to take this into consideration. So I do not really see a purpose here, but that maybe there are indeed some specimens out there that you would like to observe that are very oxygen sensitive, maybe. And in this case, what I can imagine is that you might have to seal the specimen in a microscope slide without oxygen being able to reach it, right? But this kind of protects the specimen, but there's still, of course, air between the objective and the slide. So, in other words, I do not see a big interference of the outside air. And you said air, and particles from interfering. Well, this would be kind of dust. This should not be interfering. I mean, there is not so much dust around anyway. So I would say I would probably need a little bit more information on concerning this question of what actually makes you think that this might be necessary. So maybe you have some kind of specific applications where this might be an issue. But currently, right now, I cannot think of any possible application, right? Because if you really want to exclude the possibility of air between the objective and the slide, then you have to place everything. You have to completely seal it off airtight. And don't forget one thing. Then the same thing would happen with electron, as in the case of electron microscopes. If you place a moist sample, one that contains water, into a vacuum, then the water is immediately going to evaporate. It's going to essentially boil away. So this means that you have to actually use specimens that are either completely dry or mounted in permanently mounted slides, right? So this is simply something that I would like to say here, that I'm quite sure maybe if there are applications like this somewhere, then I'm quite sure that some people would have actually resorted back to a homemade solution, or maybe universities would actually have an engineering department that actually would manufacture parts that would actually then allow you to observe specimens in a vacuum. Okay, but I would like to move on now. Also, do you have a video on obsidian? That's a mineral, right? It's a volcanic mineral. It's like a glass. I heard it's the sharpest or finest blade at microscopic level. So I suppose when it chips off, that you have it's very sharp and fine. It would be awesome to see how it looks under the microscope. Unfortunately, I don't have access to this mineral. I do assume that when you say that it's very sharp or fine, then I suppose that you are talking about the edges of obsidian when it chips off. Maybe it's possible to actually see how fine it is. I don't know if it would actually go down all the way to the atomic level or not. That would be kind of interesting. But if it's some kind of a crystalline in nature, then I could imagine. But I don't know a lot about this because obsidian is a little bit like glass, so I'm not quite sure whether it's amorphous or if it's crystalline, but that's something that I'd have to research a little bit. Generally, if you want to observe minerals under the microscope, because they are hard, obviously, in many cases they're hard, what you have to do is you have to kind of polish them down. And you have to really make sure that they are thin enough that you can observe them properly under the microscope. And this requires some grinding and polishing until the specimen, the mineral or the rock, is thin enough. Something that I have not done. Okay, so I'm in that sense, I'm probably not the best expert on this here. But I guess what you are mostly interested in is really to have a look maybe at the sharp edge of the obsidian that basically is formed when it starts to chip. Okay, so and another question over here. Finally, I have a question about isolating a specimen from the slide. Is there a way to do this with a minimal risk, or is it just inherently difficult? Also, did you manage to extract the tardigrade from under the cover glass or from the slide? Removing the cover glass isn't really an option since it disrupts the water and everything else on the slide. Thanks, Mr. Microbehunter. Okay, so again, a little bit of background info here. Basically, when you're seeing a small microscopic organism, in this case a tardigrade, a water bear, on a microscope slide, and you would like to isolate it because in many cases it's not alone, but there's lots of debris, there are other cells around it, maybe other microorganisms, and you want to actually have it alone on the slide because you want to make a nice video or take a nice picture, and you're not so interested in all of the other stuff that's floating around on the microscope slide, then it's advisable that you use a pipette, a microliter pipette. I'm using a one that goes to maximum 50 microliters and a disposable plastic tip to suck it off. Really, I just go and dip the tip of the slide into the droplet of water on the microscope slide and then simply carefully suck it off. So I basically observe under the microscope until I approach the tardigrade or whatever microorganism I want to pick off, sufficiently close with a microliter pipette tip, and then I simply suck it off a little bit. Yeah. So this is only possible without a cover glass. Removing the cover glass is not an option because if you remove the cover glass, then you're kind of losing again the specimen because maybe it's then sticking to the surface of the cover glass, and you have to kind of find it again. So this is generally not it's not a feasible idea. So what I usually do if I want to isolate microorganisms or microscopic animals, I never put a cover glass on top and just use the low power 4x objective because I need a large distance between the objective and the slide. This gives me enough space also for the pipette to pick it off. Yeah, and by picking it off, I usually rest the tip of the pipette on the slide, on the microscope slide for stability. And I also support it with my other hand a little bit. So basically with one hand, I'm holding the pipette, and with the other hand, one or two fingers, I'm stabilizing the tip of the pipette, which is also resting on the microscope slide. And this way I'm carefully approaching the tardigrade, and then I quickly suck it up and am able to transfer it onto a different slide with a little bit of fresh water again. And I keep on doing that, and every time when I transfer the tardigrade from droplet to droplet, then there are a number of contaminating cells and other debris and so on. It goes down as well, and until the tardigrade is basically all alone on the microscope slide without anything else. And this is usually how I'm getting my nice videos and pictures. Okay, so this is simply something that I would like to share with you. So I still have a couple of minutes, and I would like to now talk about a little bit about my YouTube channel here. I've been making videos like this now since January 2018, so it's been a couple of years now. And of course, the community is changing a little bit as well, and I would like to also ask you for a little bit of help here. Which type of videos do you like to see? In the past, the views have been going up and down a little bit. I've received quite a few views over my YouTube Shorts, but then those people who watch YouTube Shorts are not really interested in my other videos. I've tried to make some documentary style videos as well, which are kind of scripted without a face. And I'm experimenting with this podcast format. But if there are any specific requests that you, as a member of the community, has, please do write it in the comments section below. Would be very grateful for a little bit of support in that way. Yeah, it's been now also, again, a little bit somewhat over 23 minutes. I think I'm going to leave it at that. I would like to invite you, of course, to subscribe to this channel. And of course, also to subscribe to this podcast. You can listen to this podcast, of course, over my YouTube channel or also in YouTube Music. But for right now, I would like to wish you all the best. Happy microbe hunting as always, and see you in the next episode. Bye-bye.